Okay, all right. Welcome everyone to today's webinar, Streamlining Resource Requests, Showcasing Tools to Search and Request ADRC Resources. We have John Hepler from Wake Forest, John Gibbons from Rush University, and Aliquot from NYU ADRCs to showcase their tools. Each one of them will give the 15-minute talk, and then you'll have time for Q&A for each speaker. Please post your questions on the Q&A of the webinar. All right, John Hepler, take it away.
Thanks, Adeshna. Let me get my share my screen here. All right. Is everybody able to see the presentation there? Okay.
Yes, we can.
All right. I'm John Hepler. I'm a senior analyst programmer at Wake Forest in the Wake Forest Alzheimer's Disease Center, specifically in the data core. And today we're going to talk about our resource sharing platform we've named WakeShare. Before we get started, I'd like to give it is a truly collaborative effort, and I'd like to give a special thanks to Eric Fisher, Sharon Letchworth for helping us organize the flow of everything in the data, and then my data core colleagues Sarah Goswami and Jerry Barnes for the statistical and programming help. Without them, couldn't have had this platform. The purpose of the platform, WakeShare, was designed to help investigators both within Wake and outside of Wake Forest at other institutions to navigate complex clinical data, imaging, biospecimen, and non-human primate resources related to both aging and Alzheimer's disease at Wake Forest. Today, we might touch on aging, but we're just going to look primarily at the Alzheimer's disease side of things. Another big piece of this platform was to allow us to have a robust tracking system of fulfilling these requests, and that would help us to track and do reporting later, as well as to track proposed publications, which is being built now and is coming soon. Just real quickly, I'll go over just some of the highlights of the features. This is a separate but complementary platform from our Wake Forest ADRC EDC platform, where we capture all of the data related to our research. That is wakeadc.org, and this one is wakeshare.org. They're complementary. They're both built on customized Bootstrap 5 and JavaScript libraries and plugins for front-end interactivity. They have Adobe Cold Fusion as the middleware, Microsoft SQL Server databases, backend, and SAS for reporting and data. We have a suite of publicly available search tools for doing queries. Accounts are vetted and then assigned a secure login, and there's additional vetting for accessing non-human primate resources. We have an overview video we won't look at today. Everybody's welcome to look at it, but it's for anybody who just wants to get a quick overview of the system before they jump in. We have an Alzheimer's sample size query tool. We will look at that. The intake form is an intuitive save as you go wizard style intake form, and that will make a little more sense later, but basically there's no submit buttons. It saves your data from the minute you make the first change. If you accidentally close the system or drop out, your data is saved when you come back. And then we'll take a look on the back end, a tool for our administrators called the Fulfillment Pipeline Admin Tool. And the goal of this was to keep all communications for our requests out of emails. In the past, have been problematic where you get a lot of email trail and just things get lost. And this also allows us to keep everything in the database for tracking and reporting later. Okay. Now I'm going to jump out to the web for a live demo. Okay. Again, the domain name is just wakeshare.org. This is the home page, and just a couple of highlights. You can go into resources, and we have them divided by aging and Alzheimer's. Again, we'll just really look at the Alzheimer's and this sample size query engine and get to it by clicking here as well. There's a place where you can register, and this gets vetted, and then you have a login set up and assigned, and then you can log in through here. And then there is this WakeShare overview video, 3.5 minutes. Anybody can go back and look at that later if you would like. We had just found ourselves re-explaining the system to investigators numerous times, so we wanted to make a way to make it more efficient to do that. Okay, So before we get into the login, let's take a quick look at this Alzheimer's sample size query engine, I guess we'll say. Okay, so this is just a way for folks to go in and look through various data through these min and max ranges and these different categories to see if there are participants in the cohort that match the research they're interested in. We have a total of 684, and if somebody's looking for female African Americans between the ages of 65, 75, you see that the number automatically adjusts, and I have MCI. You can kind of understand how large a group of participants you'd have to work with if you make a request. Okay. All right. So like I said, once you register and you're assigned your login, then you can log in here. Before we get into actually submitting a resource request, this is your resource hub, where if you have multiple resources, you can come in. A red link that you can click on means it's something that you're currently submitting. It's not finalized. It's a work in progress. Once you have submitted it, it goes through this into this fulfillment pipeline where you can see where things are. You can no longer edit the request, but you can download a submission summary. And there's some mechanisms here I'll talk about in a little bit. Before we get into the intake form for non-human primate resources, by default, when you get a login, you do not have access. No one has access to non-human primate. You would just click here, give some information about yourself and briefly describing your needs for these non-human primate resources. And that is further vetted by our folks at the coordinating center. And then you're added to a group. And then once you're added to that group, there is a non-human primate subsection to our form that becomes available where you can ask for those resources. Okay. You click on submit a new request. Opens in this wizard style form with a little modal with some instructions. I'm going to close out of this. You can't move through this form without submitting the data that's required on each page. So what I'm going to do is go back to the hub and reload this one I'm working on. Okay, so you wouldn't be able to go to investigators without filling out the agreements, the intents and policies, and the everything here. When you make a change, you'll see this little icon come up, and it means that your data or your changes is automatically saved. So everything on this tab has been satisfied. This is where you list all your investigators with your study. You just click this to pop open a little modal, add their information, and they automatically populate this listing. Next is, and I'll mention, you can go through this form by clicking the bubbles or clicking next. Both get you to the same place. And you can click non-sequentially as long as the tabs are satisfied if you're coming back to edit a form. So this is study information where you put in information about your title, your proposal, your abstract. Is it internal to Wake Forest or external? Is it pilot exploratory? Details about your design and methods and your analytic plan. You can specify if it's related with a particular grant, grant number, start and end dates. Sorry, this is a little wonky because I preloaded it, but this will satisfy it. But you put in your real grant start and end dates. You list your collaborators both within Wake Forest and those outside of the institution, Wake Forest. Then you come to, it's really the meat and potatoes where you start to specify what you're looking for in this request. We're going to have more cohorts added in the future. Specifically, we're looking to add our OAIC Pepper group, but for now, it's just the Alzheimer's group. And in this page, we've broken down into subcategories where you can go through and cherry pick the variables that you're interested in, and you just select them. And alternatively, if you don't want to go through all these categories and picking them, we have this thing called the minimum data set, or MDS. It's kind of the greatest hits of variables that most investigators ask for. So you can just click that and it'll give you most of the variables that you probably need. But again, you can also go through here and specify them if you want individual ones. At the bottom, you can provide any further details that may not be included in these variables for clinical. The next tab is biospecimen. This is where you can ask for tissue, brain, CSF, serum. Again, as you check everything, it saves automatically. The volume, how much you're wanting, number of cases, your inclusion and exclusion criteria for clinical and pathologic, and a FedEx number for shipping. Next is imaging, where you can ask for MRI. And MRI is divided into two categories. Brain, you can see in these, and these have data dictionaries, so you can download them and if you're not familiar with some of these, and see exactly information related to these categories. At the bottom is MRI body, so you can ask for scans of body. Below that, PET scans, both amyloid and tau. And then CT. You can specify by region or tissue type. And then at the bottom, DEXA data. And then here's a little key at the bottom for all the abbreviations, mainly in the top MRI portion. And then, as I mentioned earlier, if you have not asked for non-human primate access, you will not see this particular section, but we have it. We'll take a peek. It's an overview about the non-human primate resources. Then you can go through and specify the species you're interested in, the sex, age ranges, if anything related to the diet, housing, other characteristics, and in major treatment areas you're interested in, pathology, and then the tissue resources that you would like to get. All right, next we go to this details. At the top, it's got a place where you can request support. If you would like data analytics support, like biostatistical support, check that. Or if you would like data management support, you can check that. And then someone from our center would coordinate with you. And then any further details regarding this request that might not have been covered through these specific subcategories. Next, you go into a large summary. I have a red message at the top here. It says, please review this summary. Make any changes before clicking submit. We do have the ability to reopen a request, but more than likely, it's much better if someone has their request finalized. So we're in their fulfillment pipeline. We're not stopping, going back and altering the resources that we're assembling. But you can, go down and review your request. Once you're happy with it, you can hit next or submit. And now this has been submitted into our system. Click here, it takes you back to your hub. You'll notice this request now is no longer editable. You can download that summary as a PDF, but you now have this fulfillment pipeline has been activated, and you just have an initial submission. All right, that is it for the intake portion. And very quickly, I wanted to show the admin side. I thought this might be of interest to centers who are trying to do something along these lines as well. These two tabs, only administrators and programmers see. So our administrators come in here to the resource requests. Let's see. Here's the one I just put in. They have this update fulfillment status. They can also click to go straight into the form if they need to make any edits on behalf of the requester. We prefer that the requester does that. So they can go in here, they get a modal, and they can set the request status. And as I said, we can check this, save the form, and it'll reopen the request so the requester can go in and make additions or changes based on some communications we have back and forth. I'll show that in a minute. Administrative review, administrative notes. Everything in green is communication tools. So approval alerts go out to our fearless leader, Dr. Kraft, and on the Pepper side, Dr. Kucheski. We're actually putting the fulfillment resources together, whether the biospecimen, clinical, imaging, or non-human primate. You can click these and it will send out all this information within the admin, plus the summary of the request to these key people so they can help begin assembling the resources. And we keep track of when that was sent. If someone is asked for support, you can go through here and actually assign who is supporting it for as a data analyst and data manager. And I'll note everything in blue is for tracking purposes. We can track total number of participants that was shared and the date. what biospecimen sample tracking was shared and the date, non-human primate, same thing, as well as appointing a fulfillment point person and any notes. Down here at the bottom, if one of our administrators or one of the people working on the fulfillment of the request has a question and they need clarification, they can type it in here.
John, sorry to interrupt. So quick time check. We have 5 questions. So maybe if you can wrap it up in the next minute or so.
Very good.
So that'd be 10 for questions. Thank you.
Very good. Long story short, this is a way it sends the requester a notification via email, and that email they can't answer. It's instructing them to come back into the system. When they go to their hub, they'll have this feedback requested. They can say, okay, and they answer it and submit it. And now that is also within our database, within the fulfillment pipeline, and it goes out to those fulfilling the request so that everybody's in the same loop and it's all trackable. I'll go ahead and stop right there.
Thank you, John. That was incredible. Lots of questions. So, I'm just going to read out the questions from the Q&A, or you can, if we don't get to all of them by 3:20, you can answer them offline. Do you collect DEXA scans on all your participants? This is from Biera Gabriel.
No, not on all of our participants. The DEXA data primarily is related to the PEPPER side of things. I think Eric Fisher is on here. I don't think he can speak, but I think we do have DEXA data on some of our Alzheimer's participants.
Okay, thank you. The next one's, can you describe the vetting process and the criteria to gain access to the data?
Is specifically for non-human primate or just in general? Well, in general, obviously, if someone asks for a login, we're going to kind of verify who they are and then assign them a login. Then once they're in, if they ask for a non-human primate, it goes to the director of our non-human primate center, who knows pretty much everybody, but it can go through, and if they don't know the person, they can understand who they are and make sure that they are who they say they are and what they want to do with those non-human primate resources, and then we'll activate them within the system.
Great. And do you have to add them to your IRB before you share data with the requesters? That's my question that I added.
No, I don't believe so.
Okay, because it's de-identified, your IRB allows you to share it broadly. Okay, great.
Yeah, and Eric, send me a message if I'm misspeaking on that.
Wonderful. Next one is from Ganesh Chan. Tentative sample size with PET would be helpful.
I'm sorry, which one is that one again?
Ganesh Chan, the tentative sample size would be helpful. So I think anybody can do that question, get the sample sizes. So you're welcome to go to the website to look at that, right? I think your website gives you the sample size.
And there are categories that we are adding to that Alzheimer's sample size calculator. So PET, I think, is definitely one of those we want to add.
Okay, okay, all right, okay, thank you. How often is the data refreshed?
To ensure completeness and accuracy of available data. So the calculator where you see a number of samples, that is only refreshed occasionally, but when you ask for an actual resource request, it is coming off of live right now data from folks who are assembling that in samples.
Okay, great. Thank you so much. We'll move on to John from Rush. And there are 3 more questions. If you can answer them online, that would be great. Thank you.
Be glad to. Thank you, everybody.
Hi, everybody. Thank you. Everybody hear me okay? So I'm John Gibbons. I'm the data manager at the Rush Alzheimer's Disease Center, manager of information systems for the Rush ADRC. And thank you for having me today. Thank you to Sudesh, Hannah, and NACC for setting this up. And I'd like to share a little bit of our data and specimen sharing systems with you. So first off, a little bit on our cohort studies. So our ADRC has 3 cores. We're the religious order study core, the clinical core, and Latino core. The Ross core goes all the way back to 1994. We have 2 additional cohorts within the RDC, the Memory and Aging Project and the Minority Aging Research Study. Across these cohorts, we have over 5,300, almost 5,300 participants, over 2,000 samples. So we do quite a bit of data over 3 decades to share. Our data sharing is something that we've always had at the RDC, and within our ADRC, it's always been part of our administrative core. It's been growing pretty substantially. We've kind of stabilized around 55 specimen requests a year, maybe 90 data requests. That's a stable size that we seem to be able to handle well. So these specimens are international and nationally across the United States, but we do have collaborators in Sao Paulo, Brazil, Vancouver, Australia, parts of Europe and Asia. So it's pretty extensive where we're sending things out to. This is kind of the structure of our data sharing and our specimen sharing. So thinking about the intakes of requests, getting good requests comes
Sorry,
from our...
John, sorry to interrupt. Are you sharing your screen? Because
Oh, do you see it?
No.
Yeah, it should be
Sorry.
screen 1 share. Oh, sorry. Oh, yeah. Yeah, I'll run through it
Okay.
real quick.
Now we
There you go.
your
See that?
screen. Yep.
Yes, we can see
Thank
that.
you.
Okay, great. So starting real quick. So here's our cohort studies. The raw study, the clinical core, like I said, about 5,300 participants, 2,000 autopsies, a lot of requests, 50 or so a year, specimen requests, about 90 data requests. It's been growing over time, but something we've been on with. We share things, across the United States and the world. And then this is kind of the structure where we left off here with our request. So the first thing starts with the intake of requests through our resource sharing hub, the request review process, tracking our DUAs. Then I'll talk a little bit about the fulfillment of our request, our specimen distributions, how we plate them, how we generate the data, and then some of the reporting tools we use to keep track of everything over time. So before we can really jump into that, I have to stress that the whole system works really because we have a central database. The central database tracks everything from our studies, all the data we collect in our studies, our unique participant identifiers, all the research data we collect, whether it's MRI data or PET data, interview or device data, it's all kind of coming back as much as possible into our database and being tracked. And that's really the basis for everything that we do. And to really hammer that home, it really is kind of the middle of everything. There's really no system that just goes out from the database. Everything's coming in and out. So we have our hub where we're getting our requests, our external requests. We have our computer variables, which we'll talk about a little bit. Those are being processed through our SAS system by our statisticians. We have all the data coming in from our laptops, which is going back out to our web-based forms and all our data collection systems, our matrices. We have an internal website where we're kind of doing some of these internal processes for our requests. And then we have our freezer system where we're tracking all of our biospecimens, what we've distributed and what we have. So our request started with our resource sharing hub. This is something that we kind of started around 2015. We had a supplement to build it. And it kind of is the basis of getting good requests and sharing good, well-documented data. So there's three parts of the hub: the documentation, the query frequency reports, and the request for data specimens. So the first part being the documentation. That's where we have all of our variables defined and outlined. You can go in there. This is just the variables page. I'm not going to do a live demo like John did, but you can take a look at this. We have them all categorized. You can look at the index of all the variables. These are variables that we've computed through our statisticians. So we've summarized and we've cleaned things. And these are really the best variables that we use internally and we suggest other people use as well. Also, we have overviews of the studies, timelines of data collection, and different sub-studies like neuroimaging, decision-making, nutrition, things like that to stack on to our other primary studies at the center. So once you get a real sense of the data that we have and what's available, the next question is kind of looking at how much data is available. So that's really where we get into these query frequency reports and what we call our dynamic reports. So this just shows the kind of the general part of this, but if you dig into this, you'll see there's demographics, cognitive status, sub-studies, different types of omics, biomarkers that we have like imaging, laboratory results, genotyping, things like that, and some postmortem diagnoses of different pathological things like that. So you can select multiple things on this report, on this webpage. You go to generate a report, and if you select 3 or more, then it will kind of give you something like this, which is kind of a frequency breakdown. It'll give you exactly what you wanted, which in this case, I selected ROS and MCI and whole genome sequencing data, and that's 133 participants. but it'll also give you all the other combinations of 2 or 3, depending on how many items you pick. I think you pick up about 8 items. It'll get bigger and bigger and bigger to the point that you really get overwhelmed, but it'll give you a lot of data. And you can kind of see where, where you're being limited by and how many people you're going to get in your data set. So once you click on the demographics report, you'll get something like this, which will actually break down those participants for you. As long we limit small sample sizes to 20, but as long as you have a few more than, as long as you have that many, then it'll give you age, sex, education. You'll get some basic mini-mental scores and ranges and some basic diagnoses to make sure that, when you're putting together this request for whatever biospecimens you want or whatever data you want, that you're going to get a reasonable sample that's going to work for whatever research you're proposing. So after you understood the data and after you understood the amount of data we have, the next step is to actually create the request. So you can go to our hub, you can look at the query frequency reports, you can view all the data. If you want to create a request, you just need to create a simple account. We do limit requests to people that have accounts, but it's pretty simple to create an account. You just put in your information. You'll create a password, it will send you an email, you verify it, and you can create a request. The last step on the request is going to be review and submit. So as long as you don't submit the request, it doesn't matter to me how many requests you create. And if you play around with the system, create data requests and specimen requests and whatnot. It's the basic kind of information you expect that John kind of presented in his study information, funding, the specimens you've requested, the procedures on the specimens, pathological, clinical, inclusion, exclusion criteria. We have a system where you pick the variables that you'd like as part of your request, and then some other information, shipping information. You can upload some PI bio sketches, and then you just review and submit the request. Once you submit the request, we have a committee that reviews the request and approves it. That's part of our admin core of our ADRC that oversees this. The chairperson reviews the request. They can either return for revisions if there's something glaring that's wrong with it, or if it's the depending on the specimens or availability or things like that. They can open it to a committee of ad hoc members. Each committee member can vote on the request. It's an email-driven system where they come back to the website, they're entering their votes. Once all the votes are collected, the request can further be sent back for revisions, or it can be accepted or rejected by the chairperson. Once the request is accepted, it becomes part of our internal system. We treat them as a project, basically. Each project can have 1 or more distributions. Each distribution can have 1 or more specimens. On occasion, we have distributions going to different collaborators, different institutions as part of the same request, or we might have distributions that happen very spread out over time. Practice tissue now, another tissue 6 months from now, another tissue a year from now. So as specimens are sent, distributions are completed, we update the state of the project. So these different status codes kind of change as the request moves through our system. This is a representation of our internal tracking system that we have on our internal website. This is where we would track our distributions. This request only has 1 distribution, but they could have many. And this is where we track our MTAs. So we would be validating requests as we're completing the material transfer agreements or the data use agreements. Then the requests would become active, and we would track the distributions. When the specimens come in, we keep it very broad on our website because we find that works well to get the information in. We can always send things back for revisions if it's unclear. Once we have it into the system, we kind of break that down into individual specimens. Like I said, each distribution can be 1 or more specimens. So we might put something like this in to track maybe 60 cases of dorsolateral prefrontal cortex tissue to this request. The system will actually look for those 60 cases, and when it finds that they've been distributed, the request that specimen will be complete and the distribution will become complete. So even though we know which specimens we're going to distribute, or we know which project we're going to distribute, we don't necessarily know which participants to distribute. So we work pretty closely with our data team, our stat team, and our data sharing coordinator to work through those things. There's a lot to be done there in terms of lining up certain specimens, certain diagnoses, lining things up with MRI for certain projects, protecting high availability, baseline, last valid specimens that are of high value. So there's a lot that goes on there, and we work together as a team. We track that in our internal website as well, but that's it's a pretty important step that can really take a little bit of time in our system. So that gets to, now we have the specimen selection, we have a good request, we know what we're going to distribute. So then we get into our fulfillment system, which is another internal system we use to try to get the right samples, the right people in a timely manner. So certain samples are relatively easy, like plasma and serum is typically one aliquot we share. We can kind of tell the lab, this is the aliquot for this person and this visit that should be shared. For other things, we might be pooling DNA samples. We might be doing additional QC steps. We might be sub-aliquoting them to only send a certain amount. So we actually have an internal system where we track this as well. All the data is coming into our main central database. So we basically say, these are the project IDs that we want to distribute to this project. These are the types of samples we want to distribute. We can specify them exactly. If it's something relatively simple and straightforward, like an individual serum aliquot of the right level. If it's something that needs to be pooled, we can go into the system. We can select samples and say, pool these together, QC these. Let's get the right quantity together. And like I said, this is all tied together because of our FreezerWorks system. So every night, our FreezerWorks database is loading into our central database. It knows exactly what we have, what's available. We can pick from that, and we can set them to be plated. Once they're plated, our lab staff can take the Excel spreadsheet of what's been plated, upload those individual aliquots back into our FreezerWorks, add what they call a transaction in FreezerWorks, which will say these have been shipped to this project, and then everything will flow through the project state system and say that that distribution is complete and that request is complete. That's kind of how everything works there. The other component to this is the data that we send out as part of every specimen project or just as a data project. So as I said, data is flowing from our study laptops into our database. We process things in SAS. We're tracking all these variables into a central database. And we're using some tools that we've created to kind of aggregate those together. We're picking a study. We're picking other data sets, whether it's in vivo, ex vivo MRI. We link them to a project. And then we pick the variables that we want. And the system will basically automatically generate a data set in Excel or CVS. And if you go to the hub and you look at a variable, that documentation per variable, if there's 50 or 80 variables in a data set, we can take that same documentation and build it into a single web page that we can send along with the data. So people have a concise set of documentation for the data that they've created. And internally, it looks something like this. This is a kind of a simplistic view of it, but you pick scaled variables depending on the study you have or a combination of studies. You could pick non-scaled variables. You can share the data in the code books, download them through this tool. And then we have a couple of reporting systems real quick in the last minute or two that I think I have. So I mentioned the project state report. As you're creating distributions for a project and defining the specimens, those are changing. So this is kind of the view we look at of this report. It would show us projects that are validating, where we're still collecting data use agreements, or we're still collecting we're still refining specific specimens. And then as those specimens are tracked in our database, the distribution states are changing to complete. The projects are going from active to inactive, and they might sit there for a while before there's another distribution down the road, or it could be completed in the future. It could be complete from that. All this data is coming to our database, so we can query everything that's been distributed across all the projects. We get the total number of distributions, the number of unique participants, and we can do that very quickly. And it's tied into the project, so we know how many unique PIs across how many projects and whatever time. We do the same basic kind of thing with the data distributions as well. When we create a data set, before we distribute that data set, we click a button on our website. Our data sharing coordinator sends it out, and it tracks all the project IDs we've distributed, all the variables we distributed, and the date that we sent it out. So we can kind of go back and figure out, these are the variables we sent to these people. If we do update a variable in the future, we could always go back and say, hey, we changed this variable. Here's a updated version of it that might work a little bit better for what you're doing. And then again, it's just, I'm presenting this, but it's a whole team that's done it. And our director, Julie Schneider, Sue Lurgans is our data stat core leader. She's been instrumental in doing this. Brian James is our new rec leader. He was instrumental in helping me put together the supplement that funded some of the hub programmers, and our data sharing coordinator, Greg, and then our lab staff, Ryan and Shabani, have done an instrumental job helping this all come together. Thanks.
Wonderful. Thank you so much, John. We have a few questions for you. The
Sure.
first one is from Joe. Do you administer your projects through something like Git?
Yes, we do. Yeah, so the hub, all the code in the hub, I didn't talk too much about that. John said he's on Bootstrap 5. I think we're still on Bootstrap 4. It's a Spring framework Java application, but all that code is managed through Git, and then Rush is on Microsoft House. So we're kind of on Azure DevOps.
Oh, okay.
Yeah.
Microsoft, cool. And the next question is, how many people are on the review committee and who was included in that committee?
So the committee is defined as an ad hoc committee through our admin core. We have everybody that's a faculty member in the RADC. We actually have a few people outside the RADC where they've contributed data back to the center. We're going to reshare data that they've been part of creating. But basically, it's the PIs of the major studies, and then an ad hoc committee of people that have subject matter expertise on whatever that request is.
Okay, great. I'm going to skip the next one because it is to all three of you, and go to the next one, which is for large studies, do you freeze data sets or just requery for every request?
We requery right now. We are trying to generate more of a standard data set that we would share more commonly. But right now our data is flowing in and basically real time. We're QCing it, building computer variables, and then whatever is there is what we're sharing.
Great. This question is for all three of you. Can you share best practices in tracking publications resulting from the data requests or material requests? And why don't you start, John Gibbons, and I'd love to hear from Alok and John as well.
Yeah, that's something we thought about building into the hub. It sounds like John Hepler's system is a little more interactive in terms of getting feedback back and forth. So we've thought about building that into the hub. Right now, we just encourage people to acknowledge us, and then we search PubMed, and we actually have a separate tool that uses the PubMed API to bring publications back into our system that we track them that way.
Great. So, okay.
And yeah, this is John from Wake. We're currently building a system. We were tracking publications in the past in REDCap, and now we're building that into this system so somebody can tie a publication to none, one, or multiple resource requests. So we know like what ties back to that. But that's still being fleshed out right now.
We at NYU are doing kind of sort of the same thing. We're using another tool to search through PubMed, and then we do request collaborators to cite us when we are sharing our resources.
Great. Do any of you put that in your data use agreements that you have to cite us and put the phone number in the acknowledgement? Okay.
It's part of our data use agreement. Yeah.
Yeah. Same here.
Excellent. And then Joe asked, do you search for the grant number? I guess that's how you track in PubMed, right? With the grant number.
Probably. I'm not sure actually, but yeah.
We do PI based as well as grant number.
Okay, great. Okay. Thank you so much. So on to our last speaker, Alok Vedvyas from NYU. Take it away.
Thank you. Let me just make sure it's in presentation mode. Thank you, Dr. Das. And it's hard to come after both Johns, but I'm going to try. So our data management and stats core teams led by Drs. Shao and Batinsky. I'm the data manager as well as the associate co-leader. And we have a pretty robust team that helps take care of day-to-day data management and biorepository functionality. I'll give you a little bit of our history so you have an idea towards what gravitated us towards the solution we put in place. But our ADRC has been in operation since 1990, but we had data from 1978 onwards. So it's changed hands. The data has gone into different versions. We incorporated the UDS, but of course, different versions of the UDS. And then we started getting into this problem where things have changed, data's changed, resources have changed, so we can track everything towards what time point did we stop doing things. So we took something from Russia's handbook. They give out a timeline on their website. So I'll give John kudos on that one. So we took it from him. We started creating these timelines where we are now, this is going on our website pretty soon in the summer update. And so this way collaborators could see what kind of data was available and what kind of data and resources are available for them going forward. So as we were working on these things, we also wanted a system that was extremely discoverable, right? So we had six goals to fulfill for a system that we could use. One was, we had all this data, we wanted to throw it at that system, and that system should not crash. It should be able to handle. It should play nice with REDCap because that's our main EDC. That's the one that's central for us, but we had other systems like LabVantage's LIMS for our biospecimen inventory. And then we wanted it to be easy to use, right? Easy to implement, low or no code, because we had research assistants who had no experience in programming. But so we wanted to automate things so they can do basic programming, basic coding, but not be burdened by learning how to code. Although we want everybody to know how to code a little bit. We also wanted the medium to be a little secure and compliant. And of course, we are research, so we are always looking for money, even when we have projects. So we wanted to be inexpensive within budget. And the last one was it should be fitting the institutional requirements. I put that it's a cold place because we actually should have used the institutional requirements first. We actually went to them after we chose our solution, and then we got some pushback, which I will talk about in a second. So we went with Tableau, which was already in our environment. We use it. It's a visual analytics platform. It was already implemented for the clinical core at our ADRC for reporting purposes. It can use multiple data sources. We can control data access as well as user permissions. But one major thing was we could not embed this Tableau instance into our ADRC website. And as everybody knows, all institutions are locking down their websites for security reasons. We understand that. So we had to come up with a solution, which was to put it on the public Tableau website. So our frequency generator currently resides on a public Tableau website, which is being fixed. And so hopefully it will come back home in the summer. So the reason for using Tableau, we've been throwing a lot of data at it. It was created for big data, so we are able. It does not crash. It's able to give you the speed. It's a robust and scalable instrument. We've been able to attach multiple data sources using APIs and also the built-in connectors, and it's user-friendly. It's low code. People don't need a lot of programming skills for it, and it's also the viz that's why it's called the visualization. The graphical representation, it's able to understand that, which kind of device it's on. So depending on that, it adjusts itself. So, and one thing I'm not mentioning here as something popping out, it's also very cost effective because it's out of the box, off the shelf, and for research community, the desktop version is free or of little cost. We were able to piggyback on the NYU servers and then get the research license for the desktop versions, which synchronizes to the public site as well as the internal NYU site. So our setup, as I said, our REDCap is our main EDC. Our data acquisition is mixed model. We're using electronic data capture, and of course, neuropsych is still on paper, so that gets uploaded, direct entry into the database. Then we're using external modules to send data over to Tableau Public as well as to internal Tableau, and then the internal one gives out the reports that has PHI. People, our clinical core is able to take a look at information there. But then the Tableau Public interfaces with the resource request form. So in order for anybody to request any resources from our ADRC, you go through our website to get hold of the resource request form, and then do a frequency search to see whether or not we have the kind of data you're looking for, or we have the end for your project. Or you can go straight from Tableau Public, look for our ADC, because historically we were an ADC, recently became an ADRC, and then come back to the resource request form. On the back end, this is how we're connecting from REDCap into Tableau. We're using external modules, so you'll see right here, the Tableau connector is there. It's being phased out because REDCap basically now is able to interact better with Tableau. They've built that functionality in, starting from 11.1 version of REDCap. On the public side, we've been using SPSS files because we use them to strip down all the PHI, because we don't want any PHI accidentally going out on the public Tableau site where people are running the queries. And that one is locked to a point where nothing can be downloaded from there except the pictures or the graphical representation. So when you go to our public Tableau site, this is what you see. Basically, we set it up based on the frequency charts based on the baseline evaluation and the most recent evaluation. So depending on where you are, you can actually pick and choose. Our brain tissue inventory is also presented there. We are working on putting up our plasma samples as well as other biospecimen information up there. This is, I think, the first, I would say 1.5 version of it. And then there's a new version coming in the summer. You are able to drop down the menus. You can currently actually look through based on demographics, cognitive status, biomarkers, and omics. But you, if you need more information, you can always reach out to us, and we can provide you with more detailed information towards what we have available. I think somebody asked John about how often we update these. So historically, we were updating it every 6 months, but because of all the new emerging technologies that we popped into our evaluation, we have actually slowed it down a notch because the data has to be QC'd a little differently. And so we wait for it before we make it available. As I said, as you're logged in here, you can download information from right here. Basically, it will just give you the graphical representation. It won't let you download any data. And then we made it into a one-stop shop. From here, you can request data or request resources. Probably we'll be changing that to request resources instead of data. So once you click that, basically our REDCap form kicks in. Just like all of the ADRCs, we're asking for the same information. You know, what kind of resources you need, what's the project title, who are the PIs, how do you, is it funded, not funded, and the grant information, so we can actually credit it back on in our inventory system. So here you'll notice as people access this, as they're filling this information out, if they've come through just our website, we do remind them that, you know, there's a way they can access a Tableau to do a frequency generation, to take a look at the sample size. And so they can just click on that link. And then as they're going forward, we provide data dictionaries. Of course, again, there's a link there. Data dictionaries are truncated to the sections that they are requesting information from, and they can be downloaded in Excel format. We're working on changing that format a little bit and providing a code book, so just like what NACC has for the whole evaluation. So data request is the same way, like most ADRCs. There's a request form that you fill out. You know, administrative core reviews it, presentations done to the executive committee. There's a notice of approval that's given out. We're unique in the way that our DMS core also has biomarker core people involved. So we actually assign a person as a liaison who can finish all the requests from you know, from the approval all the way to delivery. So what that person does is interacts with all cores for that, for the requester. So that way we can fulfill everything the way they want it. And if we have additional information or they need additional information, we are able to provide it to the person. So as we were putting all this in place, there were lots of lessons learned. So I'll just touch on a few of them right now. We migrated from a paper trail to electronic. So I would say document your data request process, even if it seems easy because you've done it on paper for years. There are always small things that you can leave out. So always document. Facilitate self-service. We realized people were asking about the same thing over and over again. So our dashboard has evolved to incorporate certain things that all researchers ask for on a regular basis. That way, it gives them the power to actually do everything before they come to you and get the information. Also centralized, right? Have a single point of entry. That's where electronic stuff, there are multiple points of entry, multiple points of exit. It's not a hindrance to have a single point of entry. It's more about metrics and to see how well your systems are responding to all the stuff you've done. Of course, communication, that's why we have the liaison who deals with the person all the way from approval all the way till the delivery of the resource. And then, of course, metrics and knowledge base, because everything adds to our knowledge and helps increase the productivity. That's how we were able to create templates for different things. As I was actually working on this presentation, somebody sat here in my office questioning me about what's a good way what's a successful request, right? So I decided to pop this in because of that. So, because I was like, okay, guys, so this is what I told them. I was like, first, communicate with us. You're going to start a project, have the data team already have a conversation with us. We'll tell you what's in there, right? Yes, we can give you all the data dictionaries, but having a conversation will give you a different kind of information from us. And give us all the context so we can give you more information. And be objective and have your goals ready. Be more focused about what is the question you want answered. Don't just have to design very thought out, more or less thought out. And be sure that you can identify the data you want, right? So don't just come up and go, just dump all the data for us, because I understand in the big data area days, everybody wants all data, but a lot of times we can't just give you all the data because we're still QCing a lot of things. And then, of course, do the reasonable time frame. Ensure that everything, if you're going to give us a data request or a resource request, which is going to take a lot of time, remember, it's going to take us time to get it to you. All the agreements that both Johns have discussed, yes, we track them on REDCap, but remember that also adds to that time frame. And have an open mind, right? So your data guys, your resource guys will actually, they might have unexpected insights. You might be looking at the problem a certain way, hypothesis a certain way. They're using the data. They have access to these resources. They might be able to guide you a little better towards what can or cannot be used. And of course, give them feedback. It's an iterative process where even after you get the data, come talk to us. We'll talk to you. Let's make sure you're reading our data right? And then things might be a little different. We can always adjust with what you want. And I love dashboarding things. So, I'll end on this one that, dashboards love to give all the bad news, but I really hope one of my dashboards can automatically filter all the bad news out. Thank you.
Okay, we are doing well on time, so there are a few questions for you. Please clarify why Tableau needs to be on a public website, I guess, opposed to a desktop application.
So NYU IT blocks embedding the Wiz within our web portal. So we had to get permissions, but before they could implement systems, we temporarily got permission to put it on the public site. So this way it's available for everybody. So we are able to give a frequency generator out to our collaborators and to researchers. And by the time that our new system comes into place.
Okay, great. I think I'll go with the next one, which is related. Do you have usage metrics on the public site traffic? If so, can you share the level of engagement you've seen?
We do. So Tableau Public does provide that information. I don't have it offhand, but it does provide us with those metrics. Last time I checked, it had about 600, 700 views after I reset it last month. But not all views end up into a data request.
Right, yeah, exactly. So sometimes you're just exploring, including me at some point. Yeah. Can you review why you chose to use Tableau and REDCap in tandem?
Well, we migrate, so RedCap, so we used to be on Visual FoxPro. This was in olden days. We've been in existence that long that our database system was on Visual FoxPro. So one way to migrate, John's nodding because, and to migrate off to a web-based portal, initially what we did was we used SQL with an Access backend. Cold Fusion at that time was extremely expensive to roll out, but then RedCap came into being, and so we migrated all our data into RedCap, which was great because it was inexpensive and easy to use. So we had RedCap in place, and then we were looking for a solution which was not too expensive, and Tableau came to be that one. So that interaction became really easy to use, and we put that in place. We are doing a lot of stuff that both Johns discussed in their systems but we're doing it on the back end in RedCap, and of course, looking into systems where we can migrate them out, because RedCap is a little kludgy with that kind of information.
Great. For everyone, what do you see as important to report about tracking the improvements or other metrics that indicate that these large systems are impactful for your external reviews, etc.? Question from Joe.
So I think those metrics are hard for us to track explicitly as data points, but the number of requests, the emails and phone calls we get to refine variables or ask about sample size, that's very real to everybody that's in the building here. The amount of time it saves. So.
Are there any specific obstacles or limitations you've seen in using Tableau for these purposes? Let's come back to Aliquot.
Not yet. We are throwing things at Tableau to see what it can or cannot do. A couple of things that we do know Tableau has a problem with is it has its own set library of graphical representations, right? The graphs, even though they're easy to roll out, it's pretty stuck. So we're looking into how we can expand that library a little bit and be able to integrate different kinds of graphs. But otherwise, no, up till now, it hasn't stumped us.
Excellent. There are 3 more questions, but we're almost reaching the top of the hour. We saw 3 different systems for tracking and fulfilling requests. Excellent data systems, and thank you so much for your wonderful presentations. A big hand to the John and Alok. I'm going to end this session with a question because so we have these individual ADRCs that are fulfilling requests, and then there is NACC, of course, who is taking everyone's data and fulfilling requests. Will there ever be a time, or is it even worth thinking about like one system that we can all think about tweaking and taking and sharing and evolving so that we have a harmonized system for making at least some of these common data requests for the UDS data elements and biospecimens? I know we don't have time to discuss that, but let's carry on this discussion on Slack, or we have a mailing list, and we'd love to hear feedback from the whole data community and the ADRC community. So any thoughts, feedbacks, please let us know. And thank you all for joining this webinar today.
Thank you, guys. Appreciate it.
Thanks for having me. Thanks, everybody.
Bye.